Hello, and welcome back to What is a Biology? I'm your host, Mr. Johnny Hopkins, and today we're covering post-transcriptional control, part 2. We'll be primarily going over RNA interference, a few different transcription factor regulation as well, and about the nucleolus and a few different RNAs, some little tidbits here and there. So primarily today is about RNA interference, and RNA interference is abbreviated as RNAi. So what does this guy incorporate? It incorporates siRNA and miRNA. Now, what is siRNA and miRNA? siRNA is small inhibitory RNA, and miRNA is microRNA. And there are about 1,000 microRNA in the cell. Typically, siRNA is made for knockdown procedures in the cell, but they are pretty much the exact same thing as miRNA. They're both about 21, 23 nucleotides in length. And the only difference between siRNA and miRNA is that the siRNA completely complements the mRNA versus the microRNA only complements about half of it, around the 10, 11 base pairs. And what happens is that you get degradation when the siRNA is present, and with the miRNA you only get inhibition of the translation of said mRNA that it base pairs to. So about 1/3 of the human genome is actually regulated by RNA interference, and these guys are made by Pol II, which they're made in about 100 to 1,000 nucleotide transcripts, which are then cleaved down into these 21, 23 nucleotide segments, which then go into the RISC complex. So what happens is that these guys get bound by an RNA base pair, an RNA binding protein, TRBP, as well as dicer, which they process the miRNA, and then they go and digest the RNA. So they digest it into a 2-turn A form, which and then it totally, with dicer, becomes, with some other proteins as well, becomes this RISC complex, which stands for RNA-induced silencing complex, RISC. So you have these RISC complexes with the miRNA or the siRNA bound. So what happens is that the miRNA is bound by the RISC complex, then some mRNA comes along and it binds to only about half of it. And from there, the RISC complex just keeps it bound. They're just bound together in that complex. And so any ribosome that wants to make it gets stuck. And it can't make it because they got this guy binding it and it can get degraded later on. However, that's not the main purpose of the RISC complex. It's just grab onto this mRNA, and keep it there until degradation eventually occurs. Whereas an siRNA, small inhibitory, the siRNA is just sitting there, the mRNA comes in, and then the RISC complex will digest the RNA, will cleave it, and then you got these fragments which then go and get rapidly degraded in the cell. So siRNA cleaves the RNA, whereas miRNA leaves it intact. But they both do the exact same thing of inhibiting translation of that specific RNA. So, as I mentioned, this is the primary technique for RNA knockdown. So you get this thing called knockout, where you just take out a gene inside the cell, which can be a very tedious process because you have to change out all the genes. Whereas in this guy, you have RNA knockdown. You can just make some siRNA that is the exact same as the transcripts, and so that gene is no longer produced, and then from there you don't have any expression of that gene, and then you see the effects of the cell without the expression of that gene. It's a less tedious process than knockout. So it's essentially inhibiting the expression by some single-stranded complementary RNA, and you have this injected. It's most effective typically though with about 100 base pair segments of double-stranded RNA. And you can also do it with some double-stranded siRNA, which will then tear apart and then go on. And so the guy that actually cleaves in the RISC complex is Argonaut, which will go through and take that 100 base pair RNA and cleave it down into the 21, 23 nucleotide segments. And you also have some siRNA that actually goes inside the nucleus, and that will target nascent pre-mRNAs, and then it will actually induce the methylation of histone 3 lysine 9, which will then cause heterochromatin to condense will cause chromatin to condense into heterochromatin and then get repression of said gene. And further, this is one thing to just quickly note, is that in the bacteria, RNA exists for about minutes. It's got a half-life of minutes, whereas in the multicellular, the metazoans, we got hours of these wiggly boys, these mRNA, existing inside our cell. And because these guys exist for hours on length, we need to figure out a way to degrade them quickly when they're not necessary, and they're just producing and producing and producing just in response to some hormone that was no longer present for the next 2 hours. So we need to make sure that we regulate our cells, and that's why this RNA, miRNA, exists within the cell. So now we're going to go on to something completely different. We got this thing called the Tor pathway, which Tor is the target of rapamycin, which is a antibiotic, which we can actually digest, which has been found and studied in yeasts by these mutants of the rapamycin inhibitor. So Tor, it's about a 2,400 amino acid long kinase, and the multicellular, cellular, and metazoans, we've got mTOR, which is just our version of Tor, and they respond to the cell signal for growth, and they activate some transcription factors by phosphorylating this guy called 4EBP, which then releases EIF4E, which causes translation. Quickly remember, IF, initiation factor. So that's the guy that gets this guy going for transcription. Yeah, transcription. So we also have this guy called Rev, which is part of the RAS family, that can actually binds to the mTOR when it's bound to GTP. Sorry, RAS is a GTPase, which are a cycle between they bound with GTP, the hydrolyzed GDP, and they go through factors with GEF and GAPs. So Rev will bind to the mTOR and then activate it, which then can actually further be regulated by TSC1 and TSC2. And if you also can have other growth factors that will phosphorylate the TSC1 and TSC2, which will then increase the Rev GTP concentration, which will thereby activate more mTOR, which will activate the initiation factor for transcription. You've also got this guy called PKR, which is a bit more related to this RNA concept, that are activated by double-stranded RNA. And they signal viral infection. So when a virus comes in, it injects a bunch of RNA into the cell, and it can usually be double-stranded, so then it will say, hey, this cell is infected, let's shut down translation entirely, let's kill this cell, apoptosis this guy. So what happens actually in the cell so that's what happens in the cell regularly, because it requires a lot of double-stranded RNA that is not self, and one way that adenovirus has actually found this is by having a 160 nucleotide viral associated RNA, and this guy actually forms a double-stranded hairpin and bind the PKR very, very tightly so it doesn't do anything else, allowing for it to just go undisturbed and make its own viruses all the time, all day, every day, until it all dies. And then you got this guy called iron response element binding protein, which has absolutely nothing to do with RNA, but we're going to cover it anyways because it's a little interesting. So this guy controls the concentration of iron and regulates the translation of ferritin, which ferritin, what essentially it does is it grabs onto the to iron in the cell and make sure they've got a steady concentration in the cell of iron, because if you have too much, bad things happen. If you've got too little, bad things happen. So you got to try and make sure that the iron in the cytosol is relatively maintained. So when you have bad, so iron response element binding protein, IRBP, will, when the concentration of low iron, the 5' UTR of the iron is a stem loop, and the iron binding protein will actually recognize the 5' bases and upstream of the gene, and then block the AUG recognition so that you do not produce any extra guys that will contain the iron, so you can get a higher concentration of iron in the cytosol. Whereas if you have high iron concentration, the IRBP will become inactive and ferritin will be produced because it's no longer blocking the AUG recognition. So we're going to talk about some fancy little RNAs here. One thing to remember is we're talking about primarily mRNA here, right? mRNA. Whereas we need to remember that's only about 5% of RNA in the cell. It's primarily rRNA, which is about 80% of the cell, and 15% is about tRNA. So, one thing to just quickly remember with rRNA is that you actually have in the genome is ordered with the 18S, the 5.8S, and then the 28S rRNAs in that sequence order, and the pre-rRNA and the pre-tRNA, those little segments actually help fold it in, and then they're cleaved, and then they go and do their own thing later on in the lifespan of these wiggly boys. So a single pre-rRNA is actually all that's necessary to nucleate a nucleolus. Nucleate a nucleolus is a weird thing to say. What that just means is it's what is necessary to create a nucleolus. Once you have a single rRNA, it creates a nucleolus in the cell. What is the nucleolus? It's the location of rRNA synthesis, essentially. And so once you've got that guy, it creates this little black dot in the middle of the nucleus, which allows for the RNA production to substantially increase. And so one of the ways that this happens is by the processing of methyltransferase onto the 2'-OH of the rRNA with pseudouridination by small nucleolar RNA, which is siRNA. Real quick about tRNA, these guys are about 75 to 80 nucleotides long, made by Pol III. Around 10% of them have modified bases, and the 5' has a variable length sequence, which are cleaved by RNase P. So after that, you've got the 3'U gets changed out with the CCA tail, which is what the amino acids, which is recognized by amino acid transferases and which stick the amino acid on it, and so it becomes an AA tRNA AA. So that's what we'll actually call a control step and make sure that everything's going okay in the cell. And so these tRNAs are actually in this long segment in of itself, and they're actually usually separated by about 14 nucleotide length introns, which get cleaved out by the RNase P, and then further regulated down on with things like that. And finally, we have something called a causal body. A causal body is a 200 nanometer to 1 micron length spherical structure, which is actually the center for RNP complex formation for the spliceosomal SN RNPs, which were covered in the previous lecture. And these causal bodies are actually made up of these SCA RNAs, small causal body associated RNA. So all in all, that is some RNA interference, as well as some different regulatory systems in the cell. So real quick, RNA interference is done by microRNA and siRNA. MicroRNA binds halfway, siRNA binds fully. siRNA causes degradation, whereas miRNA only keeps it locked in. And this is a process in RNA knockdown, which is which is a lot less tedious process than knockout, but gives the same results. You have a Tor pathway that exists. You have a PKR pathway that gets activated by the viral genes with a double-stranded RNA existing, which will indicate apoptosis in the cell. You have iron response element that will increase the binding of iron in the cell if there's too high of a concentration, or will not allow the production of ferritin in low concentrations of iron. rRNA is made in the cell, and a single rRNA is all that's necessary to make the nucleolus, the site of synthesis of rRNA. The tRNA is always post-transcriptionally modified to make sure that it gets the right, so it's able to give proper translation inside the cell. You've got these guys, Cadre bodies, which are the center of RNP complex assembly for the SNO RNPs. Oh, spliceosomal SNRNPs. So that is all I wanted to cover for today. As always, I'm Mr. Jordan Hopkins. Thank you for watching.